It's a real pleasure to introduce Eric Nestler from the Mount Sinai School of Medicine, the Icahn School of Medicine at Mount Sinai, to be exact. He's the Nash Family Professor of Neuroscience there, and he is also the Dean for Academic and Scientific Affairs and the Director of the Friedman Brain Institute. Eric received his bachelor's degree, his PhD, and his MD, and did his residency training all at Yale, so very familiar with the New Haven area. And he is a member of the National Academy of Medicine. He's a fellow of the American Academy of Arts and Sciences, and he's the past president of both the ACMP and SFN. He's the founder of a scientific advisory board chair for psychogenics and a member of the board of directors of Berg Pharma. He's the author of more than 600 publications and 5 books. And the goal of Dr. Nestler's work is to better understand the molecular basis of drug addiction and depression. And he uses animal models in these disorders to identify the ways in which drugs of abuse and stress can change the brain to lead to addiction or depression-like syndromes, and to use this information to develop improved treatments of these disorders. He's going to be talking today about some more specific sex effects and some of the work that he's been doing. On a more personal note, I was very lucky to have spent about 7.5 years at Mount Sinai myself, working with Dr. Nestler, and had the pleasure of meeting with him at least once a month in our depression club, which was more fun than it sounds like. He was a real proponent of kind of transdisciplinary work, really, kind of encouraged all of the faculty to talk from neuroscience to psychiatry and beyond. He has mentored hundreds of people, many of whom have gone on to do really wonderful things and change the field themselves. So it's a real pleasure to introduce Dr. Nestler, and really looking forward to hearing your talk. Thanks.
Thanks so much, Kate. I really appreciate that very kind introduction, and I should say we still miss you here. So thank you everyone for joining in. I was supposed to be there in person, but I guess Zoom is the next best thing in this age of COVID. So I'm going to share my screen and get started. And as Kate mentioned, I'll be speaking to you about some of the recent work that we've done looking at sex differences in depression, focusing on transcriptional and epigenetic mechanisms. So what I'll do today is I'm going to give a very quick overview of what I mean by transcriptional and epigenetic regulation. I will then illustrate how we've used genome-wide data sets to gain unique insight into the molecular basis of sex differences in stress susceptibility in rodent models, and its component resilience, and then the incidence of depression in humans. This is important because, as most of you probably know, some better than I, that depression is twofold more common in women than men. There have been some sex differences reported in the clinical features and treatment responses of depression, although in general, those differences tend to be relatively subtle compared to the whole syndrome. And we know relatively little about the molecular mechanisms involved. And if I have time toward the end, I'll also talk about how genome-wide data sets have given insight into how early life experience controls future susceptibility to stress. So this is my introduction to epigenetics. We know that a mammalian organism has about 3 billion nucleotides in its genome, a mouse and a human. Somehow that those 3 billion nucleotides, if stretched out linearly, would be about 2 meters in length. And somehow that's compacted into a microscopic cell nucleus. And the field of epigenetics briefly is focused on understanding how that incredible level of compaction occurs. So we've learned over the last decade or two that the DNA double helix is wrapped around octamers of histone proteins to form the unit of chromatin called a nucleosome. Nucleosomes are then organized in very rote ways in the most condensed form in what we would recognize as a chromosome. And this organization is very important because spans of DNA that are in compacted regions of the genome cannot serve physiological functions. For example, they can't serve as a template for RNA transcription, whereas DNA located in more open areas of chromatin can serve those functions. And for that reason, a focus on chromatin mechanisms helps us identify the genes that are affected by stress or depression. But even most importantly, from a basic science point of view, the availability of chromatin biology has made it possible, really for the first time, to study transcriptional mechanisms in vivo. Previous work had focused on identifying changes in steady-state RNA levels in brain, for example, but then any effort to understand mechanisms required in vitro studies, even though we know that what happens in a cultured cell, even a cultured neuron, is not always an accurate reflection of what happens in the intact brain. Studies of chromatin now provide that insight. And the idea is that by analogy to cancer biology and developmental biology, where certain forms of epigenetic modifications that convert one form of chromatin to another, once they occur, are permanent, the idea is that perhaps behavioral experience does something similar, induces certain types of chromatin modifications that are then stable for the rest of the organism's life, driving lifelong changes in susceptibility and resilience to stress. So I'm going to just give you a little bit more introduction focused on that level of analysis. Now the DNA are these gold stripes wrapped around these spheres that are the histone octamers forming a nucleosome, showing you the two ends of the spectrum, open or active chromatin, inactive condensed chromatin, and some information about what mediates the transition between the two. We know that in a state of activation, there are relatively open spans of DNA that combine transcription factors. These are just names of transcription factors that we've studied in the context of stress. They can bind to these regions of the genome and then recruit many other proteins to the vicinity of that gene to induce its activation. For example, a histone acetyltransferase might be recruited, which would acetylate nearby histones, providing some of the thermodynamic energy to move nucleosomes further apart. But that's just one example of many hundreds of activators and co-activators ultimately responsible for recruiting the RNA polymerase II basal transcription complex. All told, in a simpler cell system like yeast or stem cells, it's been estimated that a couple of hundred proteins might be recruited to the vicinity of one gene promoter in concert with its activation. And in a similar way, the condensation or inactivation of chromatin is equally complicated, involving many types of repressor proteins. One example would be a histone deacetylase or HDAC, which would remove these acetyl groups catalyzed by the histone acetyltransferases, along with histone methyltransferases, DNA methyltransferases, and so on. Starting at about 12 years ago, when we moved to Mount Sinai, we decided to take advantage of open-ended approaches based on the idea that we don't always know enough about underlying biology to make educated guesses. And so we embarked on what's described in this slide, is to overlay different forms of genome-wide data sets to have the biology teach us of what the most important mechanisms might be. So as illustrated here, we would perform RNA sequencing as a way to identify all the RNAs that might show upregulation. A different subset would show downregulation. And if these RNAs are regulated through a change in transcription, they will be associated with a change in a chromatin endpoint to activate transcription. That might involve an increase in histone acetylation or a change in some other type of histone activational mark or a reduction in some repressive mechanism. And we would analyze these histone modifications genome-wide through the use of what's called chip sequencing or chromatin immunoprecipitation sequencing. And we would also use chip sequencing to study these types of transcription factors and many others, which we deduced are mediating many of these types of changes. We would look at portions of the Venn diagrams where we see overlap. Now, the reason for doing this is an enormous bioinformatic challenge. Simply doing the experiments shown on this slide, one time point, one brain region, one model, would involve perhaps 20 terabytes of sequencing data. So it's an enormous undertaking, but we think the right approach, because if we rely on any single platform, that approach is associated with high false negative and high false positive rates. Whereas if we drill into areas of overlap, we can identify genes with much greater accuracy and at the same time have a lot of built-in information of underlying mechanisms involved. So I'm just going to illustrate this with a few data sets that we've generated. Now, before doing that, I'm going to emphasize up front that what I'm arguing for a syndrome like depression, which can only be diagnosed today based on behavioral abnormalities, I'm at the opposite end of the reductionist spectrum, focusing on cell nuclei and chromatin mechanisms. But our view is, by taking the most open-ended view of looking at alterations in gene expression, this will provide a template of all the genes that show abnormalities in depression or animal models of depression, including many located at the synapses of relevant brain areas that drive the behavioral abnormalities, information which we could then mine for therapeutic advances. Now, in order to do this, we have to understand where in the brain we would want to look for these modifications. This is a cartoon of a sagittal section of a human brain, identifying so-called limbic circuitry that's heavily involved in regulation of mood under normal conditions and abnormalities in mood that are seen in depression. A great deal of work focuses on medial prefrontal cortex, on nucleus accumbens, a major brain reward region, in the temporal lobe, the amygdala and hippocampus, the hypothalamus, the brain's monoaminergic nuclei that innervate these four brain regions, and many other areas of brain not shown here. Now, in order to study mechanism, however, we have to rely on animal models. And this is one of the unique challenges in the field of psychiatry, because we can never know if a mouse or a rat is depressed. And I think this is one of the major reasons why advances in the field of psychiatry have not progressed at the same rate as seen in other branches of medicine, where animal models are more straightforward. Still highly complicated and contentious, psychiatry even more so. So let me just tell you about one model that we've used, and you can take it for face value. We call it chronic social defeat stress. So what we do, and I want to credit Olivier Berton, who's now at NIDA, Nadia Tsankova, who's at Mount Sinai in New York, who introduced this paradigm to the lab. What they did was to take an inbred C57 mouse, place it in the cage of a bigger, meaner CD1 retired male breeder mouse. Fighting ensues instantaneously. We limit the fighting to limit physical contact, and then separate the mice for the rest of the day in a screen that maintains sensory connections but no more physical contact. And we repeat that process every day, typically for a period of 10 days. And at the end of that 10-day period, a normal C57 mouse developed a behavioral syndrome characterized by these symptoms. We say the mice have anhedonia, a decreased interest in pleasurable activities. And actually, I think that's something that we can measure well in a mouse. The mice show decreased interest in drinking sugar water, eating high-fat food, having sex, running on an exercise wheel, and so on. We say the animals have anxiety-like symptoms. That's a big inference, because I will never know if a mouse is anxious or not. Basically, all mouse assays for anxiety look at exploratory behavior, so reduced exploratory behavior. 10 days of social defeat stress induce hyperactivity of the HPA axis, disrupted circadian rhythms, increased addiction liability, an interesting metabolic syndrome characterized by overeating and obesity. So it's interesting that the mice eat more and get fat, even though they seem to enjoy eating less. Captures perhaps a comorbidity between a subset of depression and obesity, profound social avoidance, and a reversal of symptoms with chronic antidepressant treatment. Now, the social defeat paradigm is particularly interesting because it offers a few features that are important. First of all, not all mice develop the syndrome I just described. About 2/3 of the mice do. We call them susceptible. About the other third we call resilient. They avoid most of these behavioral abnormalities, although they still show the decreases in exploratory behavior. Most of these behaviors are essentially lifelong once they're induced. So the idea of being able to have a discrete period of chronic stress followed by lifelong changes in behavior, makes it possible to study whether whether we're able to reverse the symptoms with various types of treatments. And we've been able to show that standard antidepressants, which, as you know, work in humans only after repeated administration, also reverse these symptoms in susceptible mice after repeated administration only, although single doses of ketamine, which work in people, also work in this mouse model. And this identification of a subset of animals that are resilient is very important, we think, because we know that a majority of humans subjected to horrendous stress can remain resilient with respect to many behavioral domains. So this is an example of one of the earlier genome-wide data sets that we generated, work by Rose Baggett, who's at McGill University now. In this example, what Rose did was to take four brain regions: nucleus accumbens, medial prefrontal cortex, basolateral amygdala, ventral hippocampus. 2 days after 10 days of chronic social defeat stress, showing you genes that are upregulated in yellow, downregulated in blue, in resilient mice versus controls, and susceptible mice versus controls. And for those of you who are not familiar with looking at heat maps, each of these vertical lines is one RNA captured by RNA sequencing. And then the Venn diagrams show the same data, simply comparing the total numbers of genes up or down regulated in resilient mice versus susceptible mice and their overlap. And one can, generate high altitude hypotheses of what might be going on. So for example, when we first discovered resilience, a discovery made by Chris Krishnan in the lab now at Baylor, we thought that perhaps resilience was simply the absence of the bad effects of stress, but that's clearly not the case across brain regions. In many brain regions, and most brain regions, actually resilience is the more plastic state. Many-fold more genes are altered in resilience than in susceptibility, suggesting in fact that susceptibility itself may be a failure of adaptive plasticity. Now, because of concerns of animal models, we have tried hard to always validate what we do in mice in humans. This relies on the analysis of post-mortem brain tissue from humans who die of various causes. This is from two brain banks, one in Dallas with Carol Taminga and the other one in Montreal with Gustavo Turecki. This is work done by Benoit Labonte, now at Laval University in Quebec City. And what I'm showing you here are 6 brain regions from 100 humans, half male, half female, half depressed, half not depressed. So a small data set for a heterogeneous syndrome, but the biggest data set available for depression to date. And what I'm showing you here are the genes that are regulated upregulated in, for example, ventral medial prefrontal cortex, Brigham area 25, downregulated in the same area, and how the same genes are affected across brain regions. Same thing in females, and so on. And by far the largest, the strongest high altitude observation from this study was the lack of convergence between gene expression abnormalities seen in depressed men versus those seen in depressed women. Only a 5 or 10% overlap. We spent a lot of time bioinformatically to ensure that this was not an artifact somehow of baseline sex differences, which we convinced ourselves of, but in order to gain even further proof, we turned to a mouse model. And I'll just give you an example of another mouse model, which we call chronic variable stress, simply subjecting mice to different physical stresses every day for 21 days. At the end of the 21-day period, male and female mice develop a range of behavioral abnormalities to an equivalent extent. And what we found, similar to the human situation, I'm just showing you 2 brain regions here, medial prefrontal cortex, the homologous region to that Brigham area 25, as best as one can do in a rodent, and nucleus accumbens. And you can see, again, there's limited overlap between male and female mice after the stress. A bit more than in humans, more like 10 or 20%. On the other hand, think of this experiment. Genetically identical male and female mice were subjected to identical stresses for an identical period of time, after which point they develop equivalent behavioral abnormalities, and yet gene expression converges only to 10 to 20%. So we believe this establishes that stress in animals and depression in humans is fundamentally different at a biological level, despite similarities in the behavioral syndromes. And we have more recent data that social defeat produces similar differences between male and female mice. Now, the availability of these broad data sets also enables us to provide insight into a challenge that has been made in the field, and that is the degree to which whether an animal model can ever recapitulate a mouse, a human syndrome. So I already mentioned to you that I would never present social defeat or chronic variable stress as inducing depression in a mouse. I doubt that's possible. Rather, we are assessing the degree to which a period of chronic stress in a mouse can recapitulate aspects of the human syndrome. So for 100 years, scientists have done that at the behavioral level. That's complicated because who knows what a mouse behavior means compared to a human symptom. So we figured we could do this more objectively looking at transcriptional data. And in fact, what we found is that we can recapitulate with each form of chronic stress, and this is even adding a third form of stress, social isolation, another study done more recently by Benoit, and compared the ability of each of these three chronic stress paradigms to produce gene expression abnormalities seen in each brain region studied in the human syndrome. And each of these chronic stress models induces, roughly 25% of the gene expression abnormalities seen in humans. But interestingly, mostly a different subset for each of these models. So that's interesting to us. It may relate to the fact that each type of stress can induce a different dimension, flavor, aspect of the broad heterogeneous human syndrome, and that utilizing several mouse models is really the way to derive most data. And this convergence between stress and depression is not seen when we look at public data sets for other psychiatric syndromes, consistent with the known function of stress as being the strongest, most important risk factor for human depression. So what I'd like to do now is to give you just two examples of drilling into these data sets and providing causality. Everything that I've told you to this point has focused solely on correlations. And obviously it's important to go beyond correlations to causality, and for that we require animal models. So I'm going to tell you about a gene DUSP6 that encodes a MAP kinase phosphatase. This controls susceptibility in females only, and a very similar role for a novel long non-coding RNA link 473. First, I'll focus on DUSP6. So this work came out of the study that I mentioned earlier from Benoit Labonté a few years ago, where he found that DUSP6 expression is decreased in ventral medial prefrontal cortex, that Brodmann area 25, of depressed human females, but not in males, and found also a similar decrease in the homologous region in chronically stressed female mice, not seen in male mice. So the first step is simply to validate this in independent cohorts of humans and mice. The replication is shown in this slide, where we see a suppression of DUSP6 expression at the mRNA level in this brain region of both species, females, not males. Now, DUSP6 is a MAP kinase phosphatase, which means that if we reduce a phosphatase, we would expect to see an increase in MAP kinase phosphorylation itself. The most prevalent form of MAP kinase in brain is called ERK. It's a form of MAP kinase. And so we measured directly phospho-ERK levels in mouse and human brain in chronic stress and depression conditions, respectively. And again, what I'm showing you here is, as expected, coincident with a suppression of DUSP6 expression, is an increase in levels of phospho-ERK in female ventral medial prefrontal cortex of chronically stressed female mice and depressed female humans, an effect not seen in males. Using immunohistochemistry, we were able to demonstrate that this effect is specific for pyramidal neurons within ventral medial prefrontal cortex, and I'll come back to that in just a moment. Next step is to ask whether this effect is causally relevant in the mouse model. To do this, we used a modified subthreshold form of chronic variable stress that by itself does not produce behavioral abnormalities under controlled conditions. So we took the mice, injected them with a herpes simplex virus, expressing either a microRNA that knocks down DUSP6 or GFP as a control, or expressing GFP as a control. And what we found is that under control conditions, the subthreshold form of chronic variable stress has no effect either in female mice. I'm just showing you two endpoints, novelty suppressed feeding and sucrose preference. There were several other endpoints that we looked at, not shown here. No effect in female mice or in male mice. But when we knock down DUSP6, which by itself has no effect, the combination of the DUSP6 knockdown and the subthreshold stress results in behavioral abnormalities. In other words, DUSP6 knockdown increases stress susceptibility across these behavioral endpoints, an effect seen in female mice, no effect seen in male mice, even though the degree of knockdown of DUSP6 is equivalent in the two sexes. We can also do the converse experiment. Now we're using a herpes simplex virus overexpressing DUSP6 itself, the enzyme itself, within the same brain area, subjecting animals to the full course of chronic variable stress, now showing behavioral abnormalities in after the full course of chronic stress in both female mice and in male mice. But when we overexpress DUSP6, this compensates for the stress-induced decrease that repairs the behavioral abnormalities in female mice, not in male mice, even though the degree of overexpression is equivalent in both species. In order to gain some insight into what might be going on, we collaborated with Yan Dong and his colleagues at the University of Pittsburgh, where we knocked down DUSP6 in ventral medial cortical neurons, and then studied the electrical activity of pyramidal neurons within the ventral medial prefrontal cortex. And what we found was that there was an increase in the frequency of spontaneous excitatory postsynaptic currents in the female pyramidal neurons, not in the male pyramidal neurons. So it's interesting that you can produce the same molecular lesion, a downregulation in this one enzyme, which is seen in a chronic stress paradigm or human depression, in both sexes, and yet get a functional readout in females only. And by the way, this increased excitability of pyramidal neurons seen in a state of heightened susceptibility is consistent with brain imaging data from a number of laboratories showing heightened metabolic activity within the ventral medial prefrontal cortex of depressed humans. Okay, so that's where the study with DUSP6 is currently. We're trying to understand why it is that DUSP6 is regulated only in females and why it has consequences when it's manipulated only in females. Before saying more about that, let me turn to the other story I'd like to tell you about, which is on long non-coding RNAs. So long non-coding RNAs are a more recently discovered type of RNA that has been implicated in epigenetic regulation. These long non-coding RNAs have been implicated in transcriptional regulation in particular. They're defined as RNAs that are more than 200 nucleotides in length. They do not encode a protein, although they exhibit similar structures as protein coding genes. And long non-coding RNAs are really interesting. You know, one of the things that's so notable about them is that until the advent of next generation sequencing, they were invisible to us. So those of us who relied on DNA expression microarrays or chips, these long non-coding RNAs were not on those chips. So they escaped analysis until relatively recently. So it turns out that long non-coding RNAs are more prevalent in the human genome than protein coding genes. Very interestingly, about a good third of them have arisen specifically in the primate lineage. So they're relatively late additions to regulatory mechanisms within primates. And almost half of them are brain specific. So we thought this was an interesting class of molecules to study. This is work done by Ornette Isler, and work that was just published a couple months ago, where she took the original human data set that I already showed you and looked at the long non-coding RNAs that show differential expression across the same 6 brain regions in female and male brain. And one of the things she found is, in fact, that there were more non-coding RNAs altered in human depression than RNAs for protein coding genes, which is notable. And the largest class of that non-coding RNA were long non-coding RNAs. One can see that there are long non-coding RNAs that are up or down regulated at cross brain regions in both species, more regulation in general across female brain. And within the ventral medial prefrontal cortex, extremely little overlap, once again, between the two sexes. More long non-coding RNAs affected in female ventral medial prefrontal cortex than male, with minimal overlap. In order to gain some idea of what long non-coding RNA to study, can study several hundred at the same time, we developed this algorithm shown in the upper left panel. So first we asked which of all these long non-coding RNAs, which are correlated in expression with protein coding genes. The idea is that if a long non-coding RNA is correlated with larger numbers of protein coding genes, that long non-coding RNA may play a role in transcriptional regulation. Focusing on long non-coding RNAs that are expressed at higher levels, that are differentially expressed in depression. Notice how many more of these are expressed in female brain versus male brain. Which of these are enriched in nerve cells, and which are expressed in neural progenitor cells, for a reason that will become apparent in just a few minutes. And there was only one such gene called link 00473, about which extremely little is known. Michael Greenberg at Harvard had shown recently that link is induced in neural progenitor cells by activity, by neural activity. But other than that, studied not at all, barely at all in brain. Drilling into long non-coding, into link 473, you can see that it is downregulated in our original data set across several cortical regions in females, not males. We had a replication cohort of the same brain area, ventral medial prefrontal cortex, in a second human cohort, showing more consistent downregulation in depressed females than males. We then looked at a third cohort, not by a bulk RNA measure, but now using RNA scope in collaboration with Chunfeng Tan and Carol Taminga in Dallas, showing a downregulation of link 473 expression in pyramidal neurons selectively in ventral medial prefrontal cortex, with a trend only seen in males. So that raises the question of this very interesting novel long non-coding RNA that shows reduced levels of expression in cortical regions of depressed human females. How does one study the mechanism and establish causality of a primate-specific gene? Link 473 or any recognizable homologue is not expressed in rodents. Well, Orna took the chance that even though link 473 is not expressed in rodents, that there will be enough sequence homology across link targets in rodents for link 473 to still produce its cognate biological effects. So what she did was to overexpress it. Remember, we can't knock it down because it doesn't exist in a mouse. So she just used an HSV vector to overexpress link 473. The expression was equivalent in male and female ventral medial prefrontal cortical neurons. And what she was able to show, just look at the left panel, this is a social defeat model, chronic social defeat stress, where she was able to show that in control animals that overexpressed GFP, social defeat produced its standard behavioral deficit of reducing social interaction. Overexpression of link 473 made those mice more resilient, an effect not seen in males. Similar patterns seen in a sex-specific way across several additional behavioral paradigms that I'm not going to go over due to time limitations. We wanted to move quickly to physiology, collaborating again with Yan Dong. We asked what effect link 473 overexpression would have in the same class of ventral prefrontal cortical pyramidal neuron. She found in this case that overexpression of link 473 now is reducing both the frequency and amplitude of these pyramidal nerve cells of excitatory postsynaptic currents. An effect seen only in female brain, not in male brain. Again, even though the degree of overexpression of the link is the same. And this is consistent with our DUSP6 data. In both cases, we're seeing data that align with one another. So a DUSP6 knockdown, I showed you, increases susceptibility and increases pyramidal neuron excitability. Link 473 does the opposite. Overexpression does the opposite. It is decreasing susceptibility and decreasing excitability. Given the fact that we obtained interesting behavioral and physiological data, we turned to RNA sequencing. So in this case, we now took out the tissue that is expressing link 473, sequenced it with RNA sequencing. It was a 4x4 experiment, so we had GFP control animals, and those subjected to a full course of chronic variable stress, or those mice, female mice overexpressing link 473, control or stressed. And what I'm showing you here in the heat map is a series of genes that are induced in yellow or suppressed in blue by chronic variable stress in the mice. These are manipulations are done in ventral medial prefrontal cortex. And you can see the dramatic extent to which link 473 overexpression suppresses this ability of stress to produce these changes, consistent with link 473's primary function of being pro-resilient, a pro-resilient molecule. Now we wanted to understand what link 473 direct targets might be. In order to do that, we couldn't study rodents because rodents do not express link 473. So this is why we turned first to neuroprogenitor cells to knock down endogenous link 473 and study the consequences. Using an antisense oligonucleotide strategy, we were able to obtain a similar knockdown in neuroprogenitors, cells derived from normal females versus normal males, and then did RNA sequencing and identified sets of genes that were induced or suppressed in these male and female derived cells. And you can see many-fold more RNAs were regulated upon link 473 knockdown in the female derived cells than the male derived cells. We're very interested in studying the direct targets of link 473. the method that's used in the field to study transcriptional targets of an RNA is called CHIRP, chromatin isolation by RNA purification, somewhat equivalent to CHIP, but instead of using protein immunoprecipitation, one is using an RNA isolation method. We can identify link 473 targets now using cultured cells. These are SHSY5Y cells that are a female-derived cell line. We simply cannot do this in neural progenitor cells, let alone in human brain tissue. There is simply not enough tissue to do this. So this is an experiment that will require substantial technical advances to require us to do CHIRP in a more native system. But until that, we use this cell culture system. We were able to identify a handful of targets of link 473. We were able to show that conditions that induce link 473, forskolin, which activates the cyclic AMP pathway in non-neural tissue, link 473 is induced by the cyclic AMP pathway. You can see a roughly tripling or doubling of the number of targets of link 473 upon forskolin treatment. And when one looks at the nature of the genomic regions that are being identified, there's a high concordance of neural specific genes, including genes implicated in models of depression. We can overlay these various data sets. For example, we can take all of these genes that seem to be direct targets of link 473, which are listed here, and ask how many of these had their expression levels modified in the neural progenitor cells in which we knock down link 473, derived from males or females. You could see there's quite a bit of concordance in the female cells, not so much in the male cells. So this is where our study of link 473 is now. Again, we are interested in understanding how it is that link 473 is affecting those gene targets in a mouse model, eventually do this in brain tissue and study the functioning of those specific targets. Let me back up and comment a little bit more on these dramatic sex differences. So I just gave you two examples of genes that control susceptibility and resilience in a mouse model in females only. We actually have many more examples, and we have similar numerous examples of the converse, genes that only play a functional role in a male-specific manner. So we need to better understand why it is that manipulating gene X has an effect in one sex and not in another. That's a real mystery at this point, because the molecular constituents of these cells in the two sexes is mostly the same. Underlying mechanisms are unknown. I think there are three possibilities. One is chromosomal mechanisms, hormonal mechanisms, and differences in life experiences. I mean, I don't know, that is particularly true in humans. I'm not sure to what extent a mother mouse treats a daughter or son mouse differently. I suppose they do, but certainly in humans, life experiences will be expected to play an important role. Now, given the fact that I truly now believe, and I have the data for, and I should mention that two other groups, Marianne Seni in Pittsburgh and Matt Gergenti at Yale, have replicated our main finding that depressed men have mostly different gene expression abnormalities than depressed women. The obvious question for a psychiatrist is, yes, but why is it then that the behavioral syndrome of depression is mostly the same in men and women, and most of our treatments, antidepressant medications and other psychotherapeutic treatments for depression, are mostly the same in men and women? And I don't know the answer to that question, but here are some ideas. The first is that even though there's only 5 or 10% overlap, that is still several hundred genes that are common between men and women across brain regions, and perhaps current therapies focus on those shared mechanisms. Another interesting point is that some of the same molecular pathways are altered even when different genes may contribute to specific pathways in the two sexes. So if we go back to DUSP6, which is in the MAP kinase pathway, it is not regulated, doesn't seem to be very relevant in the male ventral medial prefrontal cortex, but we have evidence that MAP kinase signaling is similarly affected in the ventral medial prefrontal cortex of depressed male subjects, but just due to different genes than DUSP6. So these are some of the ideas that we're pursuing. Let me close by telling you just a little bit more about early life stress at just a high level of analysis, and then I'll stop in time to allow for questions. So we know that early life stress is crucial for human depression. I mentioned that stress is the strongest risk factor for depression, early life stress in particular, with a fourfold increase in lifetime risk for depression in individuals with adverse events early in life. There's one of the cardinal features of this is not so much early life stress causing lifelong depression, but rather early life stress making an individual more vulnerable, more susceptible to stress later in life. And this tends to be associated with more severe illness and more treatment resistance, and we don't understand the mechanisms. Now, there's a very large literature looking at early life stress, mostly around maternal separation. You remove a mother from the pups. But in most cases, the pups are abnormal from that early life period and remain abnormal for life. We were interested in recapitulating this feature of inducing a latent risk that only becomes apparent after a subsequent stress. So I want to tell you about a study done by Catherine Pena, who's now at Princeton, and Kate set out to do this. What Kate did was to take a normal C57 mouse. These are male mice. She looked at 2 periods during the pre-weaning period, early life. She subjected these mice to early life stress, rehoused them, and then subjected the mice to a subsequent stress. Just focus on this middle lower panel, which provides a composite behavioral score of several behavioral measures. You can see that at baseline, control mice under these various treatment conditions are the same. So any deficit is latent. Subjecting mice to social defeat induces susceptibility in a subset of control mice, a subset of mice exposed to this early period of early life stress. But the ability of this second phase of early life stress to increase stress susceptibility. These are effects in male mice, similar findings in female mice across a variety of behavioral measures showing increased susceptibility of female mice to adult stress after a period of this early life stress, which under control conditions does not produce a discernible effect. So to gain insight into this, we turn once again to RNA sequencing. I'm going to just show you this data point to illustrate that one of the main findings of this study across several brain areas examined is the finding that early life stress induces a pattern of gene expression that is recapitulating the effect of stress in the adult animal that was raised normally. So I'm comparing 2 conditions, animals exposed to early life stress, then normal, no further stress. So this is basically a permanent effect of early life stress in behaviorally normal animals, versus taking animals raised normally, but exposing them to stress in adulthood. And you can see the similarity in the patterns across brain regions in male and female mice. But once again, look, virtually no overlap between the two. So the common theme that I've been telling you about, similar behavioral syndromes but very different underlying molecular biology. Okay, so with that, I'm going to skip through the mechanisms that we have for that and just go to some conclusions. So I focus today on molecular changes in limbic brain areas. I focus today only on ventral medial prefrontal cortex. We have data on all the other brain regions that I've mentioned, governing susceptibility and resilience in mouse models and depression in humans. I focused on sex differences, showed you a bit of early life stress. We're also very interested in antidepressant action. We think that by studying these models at the level of chromatin, and I gave you just one quick example of chromatin studies looking at that link 473, that we think that looking at chromatin will provide some answers into lifelong changes in susceptibility and resilience in mouse models and in humans. And the goal now is to mine these data sets for improved treatment. So I'm just going to give you an example of how we've done this. So going back to this slide, my argument was by focusing on transcription and epigenetics, we can identify receptors, channels, and other functional proteins that might be relevant depression targets. So I want to harken back to an early study that we published by Chris Krishnan and Ming-Hu Han in 2007. This was relying on microarrays, looking at the ventral tegmental area, where we identified a series of potassium channels that showed unique induction in this brain area of resilient animals. That then led to a series of studies by Ming-Hu Han's lab and my lab that perhaps if we activated KCNQ channels, that might be a novel antidepressant. It took 11 years to do a clinical study at Mount Sinai by James Murrow. This is an open label study which should be taken with an enormous grain of salt, as all open label studies should be, nevertheless demonstrating a robust antidepressant effect of a KCNQ potentiator ezogabine, coincident with a change in activation of brain areas that are relevant to reward. So with that, I'm going to stop, unshare my screen, and I'd be very happy to answer any questions that y'all might have.
Excellent. Thanks very much, Eric. That was fantastic. I'm going to give people a little bit of time to start either chatting or raising hands to ask questions. As they're doing that, I'm going to, of course, take the prerogative to ask the first question, or I guess it's a bit of a comment, but with regard to what you were describing, where you see kind of big differences at the molecular level from females and males, and then you kind of see relatively similar diagnostic phenotypes. The outcome looks pretty similar in terms of depression itself. The sort of missing link there is the question around intermediate phenotypes or these dimensional traits. And an example in some of the work that we've done in bipolar patients, we see that they're all bipolar. Those that have had early childhood trauma histories, what we see is a very different response in males and females at the level of affective processing. So we see that, you know, the kind of very common negative bias where people attend toward negative information or overinterpret neutral toward negative is seen very clearly in females who have been traumatized with bipolar disorder. In the male subjects, they actually significantly bias away from negative information. And yet their diagnosis is the same, but it's an interesting kind of, at one level down in terms of brain processing, that there is this difference that we can see. And the best we've been able to do in the human model is, of course, to try to tie it to some of the things that we know about the physiology of the HPA axis, and that there's somewhat different compensatory kind of systems within HPA in females and males after they've experienced trauma or stress. And so I wonder, you know, if you have thoughts on that, if you, of course, you've looked at lots of these things, and it's hard to do these kinds of tasks in an animal, but just kind of thoughts on that physiological and the intermediate phenotype component.
Yeah, absolutely, Kate. Your point is really well taken. And, you know, so you could, in addition to looking at HPA axis related endpoints. You could look at brain imaging endpoints to maybe find neural substrates, as I know you're interested in. Your point is right on, because one of the challenges that the field has placed on itself now is that the behavioral endpoints of chronic stress that I illustrated today very quickly are not nearly as sophisticated as affective processing, right? They're looking at the amount of sugar water an animal drinks, looking at social interaction in a very quick assay. So what we are beginning to do, what we need to do, is to look at more complex cognitive, in particular, affective endpoints to see whether there are similar endophenotypes. So maybe female and male mice differ a lot if we looked at avoidance training, reversal learning, affect, right? All these kinds of more sophisticated emotion-related cognitive functions. So we're starting to do that. Mice are stupid compared to humans, but they can do most of these tasks, and we've had some success along those lines. So I think it's a very important avenue.
Great, thank you. I'm going to read off the one question that's currently in the chat box, and then we'll also open it up for people to just jump in as they wish.
There are also several people with their hands up on the participant list.
Okay, thanks, Charles. We'll start with the written one because it's easy, and then I don't see the hands up, but we can.
Okay, I can call on them.
Thanks. So great talk, thank you. And does the RNA sequencing data that you showed for males and females exposed to early life stress include long non-coding RNAs? If not, are you looking at differential expression of long non-coding RNAs in early life stress?
Yes, absolutely. And, you know, so the PIs in the audience can appreciate what I'm about to say, is that we publish all these data sets and the paper ends up publishing perhaps 5% of the information in the data set, and the rest of the data sit there. And I, as a PI, want a new student or postdoc to go back to a published data set and remine it. So actually the long non-coding RNA study I talked about is a successful example of doing that. So the Pena et al. Studies very much included long non-coding RNAs, and we're very interested in lifelong effects of long non-coding RNAs in stress susceptibility. However, those data sets would not include link 473 because it's not expressed in mice.
We have a question from Silke Nova. Silke, can you unmute yourself? Yeah.
Thank you very much. So very nice talk. My question is about whether you have controlled for changes using antidepressants in the mice. I possibly didn't get it. And I have a second question. Of course, the ventral hippocampus is known for neurogenesis. And the question is whether you think of, from the data you possibly have seen, is this more contributing to noise, or do you think it's actually giving you more information?
So we have looked at antidepressant response with these genome-wide approaches. We've published a few papers with standard antidepressants like imipramine, a tricyclic antidepressant, or fluoxetine and SSRI, or with acute ketamine, which was just approved about a year ago for treatment-resistant depression. And with the large data sets, one can answer questions like, to what extent do antidepressants work by reversing the bad effects seen in susceptibility versus inducing the good effects seen in resilience? Perhaps not surprising, it's a mixture of both in both sexes across brain regions, but the details are quite complicated. But we do feel that we're gaining new insight into antidepressant action through this approach. The hippocampal neurogenesis, for those of you who are not familiar with it, in mammalian species, there's the continued birth of new granule cells in the dentate gyrus of the hippocampus. It's felt to be more prominent, larger numbers of newborn cells in rodents than in humans. Nevertheless, it's been clearly shown it continues in humans in evolution as well. Renee Henn at Columbia has probably done the best work on this. And he's implicated the neurogenesis to timing effects with cognitive and emotional cognitive learning. It's possible that neurogenesis in hippocampus is pro-resilient. I think the evidence, causal evidence for that is still somewhat controversial. There is some evidence that certain, but not all antidepressants might work partly by increasing neurogenesis still further. Again, some people find that convincing, others are more skeptical. I think there's more work to be done. Our data don't really address that question.
Okay.
Question from
Will Renthal, you have your hand up, and then I know David Silverswag also has a question. So let's say Will first.
Let me just say that Will Renthal was a graduate student in my lab about 100 years ago in Dallas. He may look young, but he's about 60 years old.
That's right. These are the best. Those are the best years of my life, Eric. Thanks for
Good answer.
This talk. All right, a couple of quick questions.
Yeah.
I guess one of the things that I was just curious, since we philosophized a lot about back in the day, and I'm sure we're still philosophizing about, and I'm curious your current thoughts on, is whether you think all of these changes in chromatin are causal or reflective of circuit changes. And also the second quick question is, I mean, you've identified hundreds and thousands of genes that are associated with different states. And I'm curious if with regard to depression, and I'm curious if any of these genes have been implicated by the recent depression GWAS work.
Yeah. So, happy to answer both. So the first question, for those people who are not epigenetic aficionados, may not be familiar with the controversy in the field that Will alluded to, and that is when a histone gets acetylated, for example, does that histone acetylation contribute to an opening of chromatin and binding of Pol2 and gene transcription, which is the way I presented it, or rather, does a transcription factor bind to a gene, and the acetylation of a histone is simply a concomitant of the binding of Pol2 and transcription of the gene? I think it's a somewhat semantic argument that our colleagues in the field are having. And as you know, Will, I've told you many times, I believe I'm a scientist, not a philosopher. So I always prefer empirical data. And so the empirical data that I have is that when we use locus-specific genome editing, which you had originally keyed us in on years ago, so that we can go into a nerve cell in an intact brain, bind to only one gene, and increase histone acetylation only at that gene, we can induce transcription of the gene. So to me, those data say that the epigenetics are also causal.
Right, but even upstream of that in the circuit, let's say if the neuron is being fired more because of a circuit-wide change that is perhaps upstream of even transcription, just the activity of that neuron, do you think that that may be the primary change, some synaptic protein or hormonal effect that actually isn't directly acting on the genome, but rather changing it secondarily? It is more of a marker of an abnormal circuit rather than a causal change of the circuitry.
Right. So you're saying not even that a change in histone acetylation alters levels of that synaptic protein, but it's somehow creating a nuclear mark that persists as a form of molecular memory or something.
It is a secondary reflection of a hyperactive circuit.
I mean, that's certainly a possibility, but there's no data for that yet, I would say. It's something that would be something looking at. The second question is also a really important one we've been looking at. As you may know, that the heritability of depression is relatively low compared to other neuropsychiatric syndromes. It's only about 35%. It's in order to identify GWAS, genome-wide significant GWAS hits for human depression in a diverse population, it was necessary to study something like 400,000 people and 23andMe. So we're just beginning to get the first few dozen of those hits, but we are seeing overlap of some of the hits that we have been studying. Not the two I talked about today, but some of the others.
Great. Awesome, thanks.
Sure, thank you, Will.
Okay, now David Silverswag, you had a question?
Eric, I just want to first add my thanks for your taking your time to be with all of us at the Brigham today.
My
It's
pleasure.
really a privilege for all of us who look to you as a beacon in the basic behavioral neurosciences. So
Thank you.
Thank you.
I appreciate that, David. Thanks.
My question is, it's more of a comment, I guess. What I love about your approach that you've described today in a very rigorous molecular way, pushing and pulling different parts of the system, and I think is really important for psychiatry and for understanding depression and resilience there, to it, is that you are using probes, behavioral and otherwise, of both negative affective valence, aversive stimuli, if you will, and stress, and positive and reward-based circuitry. And as we know, both are involved in depression, anhedonia being more linked to the latter, of course. And yet those, and the former is more, extended amygdala-centric, but still very much involved in salience processing throughout that whole system. And what's neat is that you then focused in on the subgenual cingulate and mentions James Murrow's study and in terms of the accumbens, et cetera. We have found sexual dimorphism in the subgenual cingulate activity in an instructed fear paradigm that suggests greater vulnerability in women, et cetera. And as you know, the orbitomedial prefrontal cortex is set up to sort of orchestrate the yin-yang of that sort of motivational valence system that's accumbens-based and the sort of aversive system, although that's a gross oversimplification and dichotomization that doesn't fully hold, that's more sort of amygdala-centric. And so therefore is a key, sort of regulatory modulatory area, and therefore a target for all the things that you mentioned. So the fact that you're zeroing in on that region and have these beautiful findings and potential, novel, non-Me Too, molecular mechanistic therapeutics, and that it's in this region that's involved in the toggling and both, of both of those systems, those interrelated systems, I think is just really exciting and where the field needs to go.
Well, thank you. I appreciate that a lot. And I think your comment really dovetails with what Kate said earlier, in that looking at the cognitive aspects of emotional learning on both valences is where a lot of the activity might be for us to gain better insight into the net functional consequences of these molecular changes we're looking at.
Yeah, and we also see changes across the menstrual cycle as well in these systems and functions. And so, of course, that represents another outgrowth that's going to be really exciting.
Yeah. I didn't mention that when we do our studies on female mice, we typically focus on pro-estrus females to control for the estrus cycle, and we see different things at different estrus phases.
Excellent. Okay, are there any other hands up? Oh, I've got one more.
Oh, I think Anna, you want to say something? Hi, Anna, good to see you.
Good to see you. Thanks so much for being with us and sharing your data. Really fascinating. I have a number of questions about, first of all, about the low non-coding RNA, but I will try to be brief. So, you showed that the effect, the phenotypic effect that you are seeing is in trans. You are overexpressing this RNA in mice, and you are observing the effect, suggests that it's in trans. On that side, you showed, one of your slides showed this RNA hybridization with sort of really local expression, nuclear, I believe, if I remember correctly, which leads me to think that there could be some cis effect or in-cis effect.
Yes.
So I'm wondering if you try to explore it, or you really think that the entire effect is in trans, and if there is some sort of this element to it. Then another question is regarding the genes related by this long-coding RNA or bound by the non-coding RNA. Do you see any association with X chromosome or chromosomes gene, for example?
Yeah, you know, so Anna, your audio was breaking up a little bit, but I think I heard the gist of it. So I agree with you completely. We're interested in both trans and cis effects of the long non-coding RNA. And for those of you who are not molecular aficionados, trans would be more like the long non-coding RNA is affecting a nearby locus, whereas the cis, it could be affecting any locus in the genome, even on a different chromosome. We'd be interested in both. We have very little insight right now into which predominates. We also don't know whether the functional effects of link 473 are mediated through interactions with nucleotides, nucleic acids, RNAs or DNA, or with proteins. And so that is another major interest we have of trying to identify the direct binding targets. And it's an illustration of how little is known about most long non-coding RNAs, because we still don't have that information available for a majority of them.
So association with X chromosome genes or Y chromosome genes, do you?
To my knowledge, none of the binding targets identified by CHIRP are on the sex chromosomes. You know, with sex difference studies in rodents, I think human studies have been more open. Sex difference studies in rodents have almost all focused on hormonal effects. You know, Catherine Dulac, you're at a distant institution, meaning Cambridge, has turned our attention more recently to the sex chromosomes directly. But I think we need to pay more attention to sex chromosomal differences that might be governing much of these differences. Looking at 3D chromatin structure in a male nucleus versus a female nucleus and so on. I suspect a lot of the regulation might be at that level.
We have a question in the chat box from Madeline Bailey. Madeline, do you want to unmute yourself, or should we read it out?
I can read it, whatever is easier.
Yeah, go ahead.
Yeah, I just had a question more about the immune system and how that could play into this. I've just read like recent papers about how the immune system plays a role in sex differences in the brain, and was just wondering if you think the immune system could be involved in this and the changes in the prefrontal cortex and kind of how that links into the story.
Yeah, absolutely. It's a very good possibility, one that we have not looked at directly yet. But I'll say that Scott Russo, who's a professor and a close colleague of mine at Mount Sinai, has focused on immune mechanisms in chronic stress models in mice and in depressed humans. Some very interesting data. So for example, he's been able to irradiate mice and demonstrate that certain cytokines coming from the bone, coming from the periphery of mice contribute to stress susceptibility or resilience mediated by the effects of those cytokines in the brain, including prefrontal cortex, nucleus accumbens, and other regions. So we think that immune mechanisms are really robust. This is leading to other clinical trials that are being done at Mount Sinai and many other places around the world, looking at humans, more women than men, who have evidence of a hyperinflammatory state systemically, and whether they might benefit in terms of depression with any of a number of anti-cytokine or cytokine receptor antibodies that are available for peripheral treatment. So, I mean, it's a robust area. Whether that whole dimension of work is related to what I talked about today, we just don't know.
Perfect, thank you.
We have a question from Laura Holson as well. Laura?
Yep. We did have Scott Russo come and speak with us last year.
Oh,
for this.
good. Excellent. Excellent.
So you got to hear some of his work, definitely. Laura, go ahead.
Yes, thank you, Dr. Nestler. This was a beautiful talk.
Thanks.
I'm someone who does research in humans along these lines of inducing stress in folks with major depressive disorder. So a lot of this is of interest to my lab. And a question that I like to ask people in the field, especially who do the animal studies, is what do you think is driving the differences by the type of stress? So the chronic defeat versus social isolation versus chronic variable stress. I'm just really curious about your thoughts about what drives differences in the outputs of those stressful experiences.
Yeah, you know, I'm only laughing because I'm a psychiatrist, maybe not a very good one, but I'm a psychiatrist who has spent a portion of my career, or let's put it this way, I was a psychiatrist trained to try to understand the psychological meaning of events in a human's life, right? To help answer your question. And, you know, to a certain extent in humans, we can perhaps think we have insight into that question. When I think of doing that in a mouse, think of how hard that is to do in a human. And then I think of how impenetrable it would be to do in a mouse. So I'll just give you a couple of examples. So we have social defeat stress in male and female mice. In both cases, the defeater, the mean mouse, is a male. So is there a psychological difference between a mouse getting beaten up by a fellow male or if a female by a male? Certainly anthropomorphizing, there'd be an enormous difference perhaps. I don't know, I have no idea what that's like ethologically for a mouse, right? Put it one step further, now I have a mouse and I'm subjecting it to different types of physical stresses, nothing particularly bad. You know, putting it on a shaker that moves like this, so that's not so disruptive, although it's discombobulating, I'm sure. Mice can be held by their tail. It's stressful, but not so bad, you know, things like that. Versus depriving a mouse of other social targets, of basically a mouse that's living alone by itself without other mice, versus a mouse that now is placed in a cage of a bigger mouse and is now afraid of getting physically injured. I have no idea of how those three things are perceived across the mouse, right? It's just becomes really difficult. I don't know if, I mean, maybe other people would have a thought about that.
No, that's great. It's always interesting to hear people's thoughts, so I appreciate,
appreciate
it.
You know, I think that what perhaps a better way to look at mouse models than recapitulating a human syndrome, whether it's depression or cancer or heart disease, because I think maybe similar problems occur across organs, is to use the mice to study mechanisms, as to gain insight into mechanisms. So if there's a finding of a gene in humans and we want to know what it is and what pathways we should look at and what other genes might serve as a good drug target if we want to influence that gene, I think that's what mice are good for. But the argument of whether what a mouse or rat is experiencing is similar to a human may just be too difficult for us at this point. I mean, I'll mention one other thing, which I think is interesting work. And people record from now a dozen brain areas of a mouse. People can do functional MRI on a mouse, although the voxel size becomes problematic in terms of resolution. But the cellular resolution of recordings from multiple brain areas of a mouse that's walking around and behaving is, you know, quite sophisticated now. People like Kafjarasa, Vikas Sohal, and others. People are now providing like neural circuit maps of the brain as a mouse is doing something. So one might even begin to look at different levels of homology, not only the type of molecular homology that I was talking about today, but what if there's a circuit homology to some of the aversive learning, affective learning questions that David and Kate were talking about? So that might be another way to gain some very high level inference of homology of function.
Mm-hmm.
Just a thought.
Thanks very much.
Sure, thank you.
Hi, this is Charles. I have a question, and thank you, Eric. This was a fascinating talk. I'm curious about the resilience in the mice. I think you mentioned that about 1/3 of the mice show resilience, 2/3 show depression. Is that a sort of bistable response? And what do you think triggers, what causes them to go one way versus the other?
Right, so that is something we're extremely interested in. I should emphasize these are genetically inbred mice. Yeah. So it's unlikely to be any genome sequence difference because the differences are so robust and common, it would be unlikely that minor genomic noise would contribute. So we are very interested in whether, the hypothesis is that just random events during development lead to the differences in function. You know, the mice are raised the same way, they're the same mothers every mother gives rise to roughly the same ratio of resilient and susceptible. So yeah, we think it's just one of these stochastic events during the development that would be a third mechanism by which a species generates diversity. So one being genome sequence, second is environment, yeah, and the third being just randomly the same genome giving rise to different susceptibilities and resiliences to make sure that the species can survive whatever the environment poses.
Yeah, yeah.
That's just hypothetical.
Right, right. Is there a sex difference? You may have said this and I may have missed it. Is there a sex difference in the genes that are induced in the resilient mice? You showed that there's a surprising lack of overlap in the depressed mice, but...
Yes, yes. The basis of resilience looks as different between the sexes as the basis of susceptibility.
Yeah, yeah. Fascinating. Great, thank you.
Okay, are there any other questions for Eric? We're coming up on 5:30, so I'll just take the opportunity to thank you again. It's a pleasure seeing you again today after spending 5 hours with you yesterday via Zoom. And hopefully we see each other again soon. It was a wonderful talk. I'm sure everyone got a lot out of it, and a real pleasure to have you visit.
Thank you, Kate and Charles. Thank you so much for having me, and thank you everybody for listening to me.
Right.
Have a great evening.
You too.
Bye-bye.